So Mandy Peffers is our speaker today, and I'm trying to find out how I can get that's better. So, and you're going to speak about the ribosome, cartilage, and osteoarthritis. And I will follow the chat. So please put your questions into chat, and we'll field those at the end as we're going along. Thank you very much.
Brilliant. And thank you. Hi everyone, this seems really strange that I'm speaking to a screen when I'm actually speaking to real people, but I'm really glad of the opportunity to talk about some of our work. So as you probably know, I work in the Institute of Aging Chronic Disease. My research group in the photograph on the left, they consist of a mixture of PhD students. Jake, second to the left, is a sandwich student, vets who are doing PhDs, vets that are postdocs, and scientists. So the outline of my talk, I'm going to talk generally about osteoarthritis in case some of you guys aren't in the OA field, and particularly what's happening in cartilage. So that is the tissue that I investigate. And then the role of the ribosome in chondrocyte homeostasis, and particularly two molecules that are involved in that. One is involved in ribosomal RNA precursor processing, and that's called SNORD3 or U3. And another one, which is an example of a canonical SNO-RNA, and in this case it's SNORD96. And these SNO-RNAs are involved in post-translational modification of ribosomal RNAs. So why are we interested in osteoarthritis? Well, I think we're interested in age-related diseases because, as you can see from this graph here, with time we are living longer. And it's fine living longer, but we want to live longer with a healthy lifestyle. When we retire, we want to be able to enjoy that retirement. So one of the age-related diseases that we are susceptible to, and most of us will be susceptible to osteoarthritis in at least one joint, is osteoarthritis. And this is an example of a joint that's involved in osteoarthritis, and that's the knee joint in people. And you can see on the right hand side we've got some histological slides showing normal articular cartilage on the left with a high proteoglycan content and a nice smooth surface, to end stage osteoarthritis where you've lost that proteoglycan and you've got fibrillation through the whole of the articular cartilage. So the problem osteoarthritis is a joint problem. It's not just a cartilage problem. And you can see from the picture on the left-hand side, there's a number of tissues that are involved. So the articular cartilage, the underlying subchondral bone, the ligaments, the muscles to some extent, and the synovium itself. And on the right-hand side is a picture of some of the tissues that we work with. And these are parts of human knees following total knee arthroplasty. So what is the challenge of osteoarthritis? Well, osteoarthritis is increasing in incidence, and the age that we get osteoarthritis is reducing, and it leads to pain and joint stiffness and physical inactivity. And this physical inactivity can cause social isolation and depression. Physical inactivity itself leads to a 9% increase in the risk of premature mortality. And this is due to diseases such as type 2 diabetes, cancer, and coronary heart disease. So physical activity is really important. And it's important because there's strong evidence of lower rates of all-cause mortality, heart disease, blood pressure, stroke, metabolic syndrome, type 2 diabetes, and some sorts of cancer. But also importantly, physical activity reduces depression and falling in older people can lead to fractures. Plus, physical activity has shown strong evidence of a higher level of cardiorespiratory and muscular fitness, healthier bone mass and composition, healthier bone, and higher levels of functional health and better cognitive function. And these are what we strive for in older age. So what is the solution to this problem? Well, treat the osteoarthritis. And it sounds simple, but actually there are no disease-modifying drugs that are available, and treatment is principally pain relief or a new joint. So early onset osteoarthritis, what can we do? Well, there's oral drugs that can be given, so non-steroidal anti-inflammatories, certain types of physical therapy. And then as the disease progresses, the treatment options may be intra-articular steroid injections, intra-articular viscosupplementation, advanced pain medication, or such as tramadol, for instance, and end stage, it's a new joint. And that's fine if that joint can be replaced, but one, it's expensive, and two, lots of these need revision. So doctors aren't really willing to give you a new joint until you really are at end stage and you're older, because they don't want you to have a new joint for 10, 15 years and then to have another one. So the aim is to try and stop osteoarthritis earlier. So currently we've got this wait and see approach with pain management until the joint is worse enough for a new joint. And as I said, there's no disease modifying drugs available clinically. There's a limited number of preclinical and experimental disease modifying drugs which are in trials. And these directed treatments of disease of cellular aspect at OA cartilage is underappreciated. So actually understanding what is happening at a cellular level in an OA joint and trying to develop treatments for that. So what is actually happening in articular cartilage and in chondrocytes in osteoarthritis? So this is a nice picture of cartilage. On the left-hand side, you can see the different layers. So the superficial, middle, and deep zones, and the chondrocytes change in their appearance and in their phenotype between those three zones. On the right-hand side, you can see the collagen pattern within that cartilage. And cartilage itself is mainly connective tissue. So 1% of the cartilage is cells, and within cartilage is only a single cell type, and that is called the chondrocyte. So that extracellular matrix is made up of a number of constituents: glycosaminoglycans, proteoglycans, water, the cells themselves, and proteins. And the major proteins in articular cartilage are type 2 collagen and aggrecan. But it must be remembered that OA is a whole joint disease, and I'm only going to be talking about a few molecules and a few ways that we could maybe change the chondrocyte, and that's within the cartilage. But remember that the OA is a whole joint disease, and there's lots of these different tissues which are impacting on each other. So within the joint, I've described the tissues there, but you've also got to think about the type of joint. So in a knee joint, you might also have meniscus and your cruciate ligaments. The synovial fluid itself within the joint changes in osteoarthritis. So what is actually happening at the cellular level? Well, we get a phenotypic imbalance in the chondrocyte, and the chondrocyte changes from being a nice, healthy articular chondrocyte to a more hypertrophic chondrocyte. And what does that mean at the gene expression level? Well, the graph on the right-hand side shows you different types of genes and their expression in non-OA versus OA cartilage. And so you can see that in OA, in the chondrogenic genes, we get a reduction in collagen 2 and aggrecan. These are the most important proteins in cartilage, and a reduction in SOX9, which is the major transcription factor in cartilage. On the opposite side, we get an increase in hypertrophic genes. So for example, collagen 10 and RUNX2 increase. We also get an increase in proteolytic genes such as MMP13 and ADAMTS5. And at a simplistic level, you can think of those as scissors cutting up the cartilage matrix. And we have an increase in inflammatory genes such as COX2 and IL-6. So interestingly, although the cartilage only has a single cell type, the chondrocyte, recent work has shown that there are subpopulations of chondrocytes. So this study was published in 2019 and is a single cell sequencing study. So what they did, they took OA patients and they removed the cartilage. They extracted the chondrocytes from this cartilage, and then they did single cell isolation, sequenced it using single cell sequencing, and then looked at the phenotypes that were seen. And they found 7 different subpopulations of chondrocytes, and they're listed in this picture below. And obviously this is recorded and you'll be able to have a look at these slides later if you want to go back and have a look at this paper. But you can see on the heat map on the right hand side, we can see the genes that are predominantly influencing the phenotype of those different subpopulations of chondrocytes. So I'm now going to talk about ribosomes and just give you a bit of background of what a ribosome is and the areas of ribosomal research we're undertaking at the moment. So how did this all start? So my PhD was looking at proteomic and transcriptomic changes in equine cartilage in aging. And towards the end of my PhD, Pete, who was my supervisor, suggested I do an RNA sequencing study. So this was back in, I think I did the study in 2012. So I took young equine cartilage, old equine cartilage, and did RNA sequencing. And the paper is here on the left-hand side. So within the transcripts that I found differentially expressed in aging were these classes of non-coding RNAs called snRNAs. And I presented this work in an oral presentation at ORSI in Philadelphia. And at that meeting, there was another researcher from Maastricht University, Tim Welting, who some of you will know, and he has come and spoken about his work at our institute. And he came and spoke to me afterwards because he was interested in these snRNAs. And the reasons he was interested is because he'd studied a particular snRNA, RMRP snRNA, because it's mutated in a disease called cartilage hair hyperplasia, a type of dwarfism. And these photographs show you patients with this disease. And so we started a collaboration. And we thought we'd actually look at snRNAs in humans and seeing globally, do they change in osteoarthritis and do they change in cartilage aging? So why is this important? Well, it said that cartilage is made up mainly of this extracellular matrix, and the extracellular matrix is made of proteins, and proteins, of course, are made by the protein machinery called ribosomes. But actually, if you look at the literature, the current literature in osteoarthritis or cartilage development and protein translation, there aren't very many papers. There is a group in New York who've published a bit of work, these two papers here, and then Kapoor's group in Canada have also done a bit of work on this. So I'm just going to explain a bit about the ribosome and the parts of ribosomal processing that potentially can change and influence disease and where we've gone with this research. So the ribosome is made of proteins and ribosomal RNA, and the ribosomal RNA is made in the nucleolus within the nucleus of the cell. And there are, in the eukaryote, there's 4 ribosomal RNAs of importance: 18S, 5.8S, and 28S. And these are produced by 47S pre-rRNA here, and that is polymerase 1 driven. And another rRNA, 5S, which is polymerase 3 driven, is also produced, but in the nucleus rather than nucleolus. So this 47 pre-rRNAs process are broken down into these main constituents here, and that process is called rRNA processing. The ribosomal RNAs then undergo modifications. So those modifications are chemical alterations to certain nucleotides in certain positions. And these changes are due to snRNAs. I'll go back to that in a minute. So the ribosomes themselves are transported from the nucleus into the cytoplasm, and we have the 40S and 60S, the small and the large subunits. And they, along with your messenger RNA, which is your template, amino acids and transfer RNAs make your proteins. So going back into the nucleus here, you can see we've got an example of a messenger RNA, and you can see here we've got snRNAs. And snRNAs are predominantly within the introns of the host genes, and most of those host genes are ribosomal related proteins, but not all of them. And so they are produced as a consequence of mRNA processing. And those snRNAs, along with additional proteins, cause these modifications that I've just talked about in ribosomal RNAs. So they're really important. So potential areas that change in disease or in aging are your rRNA, your ribosomal proteins themselves, your mRNA and your SNO-RNAs, and they can all have influences on the ribosome and consequently on the proteins. And in osteoarthritis, we have in the cartilage an imbalance between protein anabolism, so making the protein, and protein catabolism, breaking it down. So this is one of the reasons we're really interested in ribosomes and SNO-RNAs. So the talk today is going to focus on two areas of influence on the ribosome. One is ribosomal RNA processing here, and the other is ribosomal RNA modifications, which is caused by SNO-RNAs. So this is another picture just to show you how we could potentially influence the ribosome. So here we've got variations in stoichiometry of the ribosomal proteins of the ribosome, changes in ribosomal proteins, and ribosomal protein modifications. The heterogeneity of ribosomal RNA can be due to alternative ribosomal RNAs and modifications in ribosomal RNAs. There's heterogeneity of the translation factors, such as eukaryotic initiation factors. I'll talk about those in a minute. And also in the heterogeneity of tRNAs. So within our group, we've done some work on modifications of rRNAs, and we've also seen that you get a change in eukaryotic initiation factors in osteoarthritic chondrocytes. And when you change the expression of some of these SNO-RNAs, which are involved in post-translational modification of rRNA. And we've also started a collaboration with a group in America looking at tRNAs, because in our sequencing data, we are also picking up tRNAs, and now we're also looking at for tRNA fragments, which may have an influence on OA. So just to break it down a little bit, on our sort of expedition into chondrocytes and ribosomes, these are the ways the ribosomes can be influenced. So ribosomal DNA transcription, ribosomal RNA precursor processing, ribosomal RNA post-translational modifications, so the modifications caused by the SNO-RNAs, and ribosomal protein composition. And the consequences of these are preferential translation, translation initiation, translational fidelity, how accurate its translation is, and translational capacity, how much their proteins can be produced by the ribosomes. And all these can influence cell fate and cell phenotype, and cell phenotype change is so important in osteoarthritis. So the first thing I'm going to talk about is our work on ribosomal RNA precursor processing and its effects on translational and translational capacity. So the SNO-RNA that I'm going to talk about is involved in ribosomal RNA precursor processing, and it's called U3 or SNORD3. Here on the left-hand side is quite a complicated picture of how we produce 5.8 18S and 28S from 47S pre-rRNA here at the top. So there's lots of proteins and factors that are involved to get the mature ribosomal RNAs, which are required for the ribosome to work. And so one of the molecules that does this is U3, and U3 is a SNO-RNA. And U3 works on this diagram here by processing of 18S. It's associated with a number of proteins, so it doesn't work alone as a component of the small subunit and is essential for 18S ribosomal RNA production. It doesn't work alone, it works with these proteins in conjunction, and this is a nice EM picture of the terminal knobs with the U3 containing small subunit processosomes. That's another hard word to say. So how did we get interested in U3? So the first study that I did with Tim was a small sort of pump priming study using a microarray, and it was a microRNA microarray, which also had probes for SNO-RNAs in it. And we did a small sample size. We did 6 young normal cartilage samples and 6 old OA cartilage samples. both from knees from humans. And we found that there was a reduction in U3 snow RNA in this microarray. So we also then looked at the difference between non-OA and OA chondrocytes, and we measured some other genes. So in conjunction with a reduction in U3, the OA chondrocyte phenotype is you have a reduction in collagen, aggrecan, SOX9, an increase in these hypertrophic markers, collagen 10 and RUNX2, increase in these proteolytic mRNAs, and an increase in inflammatory markers. So the expression of U3 in chondrocytes from OA cartilage was reduced compared to non-OA, and this is accompanied by a distinct chondrogenic, hypertrophic, proteolytic, and inflammatory phenotypic changes. So next we had a look in some mice knees. So this is a destabilization of the medial meniscus model, post-traumatic model of osteoarthritis. And this model was run for 8 weeks, and then we used in situ hybridization using U3 probe to determine where U3 expression was within the joint. And we found that there's a reduction in U3 in weight-bearing area of the joint and in the menisci, but U3 was unaltered in the non-weight-bearing area. And then we looked at U3 SNOA expression when we treated normal articular chondrocytes with OA synovial fluid. So the graph shows 4 normal patients, so P1 to 4, treated with 20% OA synovial fluid. And where it says N equals 6, that's 6 pooled donors of synovial fluid from the OA joints of end-stage total knee arthroplasty patients. So we saw when you had normal chondrocytes and you treated them with OA synovial fluid, there was a reduction in U3. So most SNO RNAs are within introns of protein coding genes, but some of them, such as U3, has its own promoter. So we wondered if the changes that we were seeing here, a reduction in U3 expression due to treatment with OA synovial fluid, was related to promoter activity. So we made a U3 promoter, and we were measuring luciferase expression, and we treated the patients with OA synovial fluid and found that we'd had a decrease in U3 promoter activity. So OA synovial fluid reduces U3 expression by regulating U3 SNO RNA gene promoter activity. So then we thought, well, we wondered what non-OA synovial fluid would do to the expression of U3 in chondrocytes. So the graph here shows the control, which is treating with sodium chloride, and then treating normal human articular chondrocytes with either OA synovial fluid, where you can see you get a decreased expression in U3, as shown previously, but when we treated with non-OA synovial fluid, so this is these are both from pooled samples of donors, we saw an increase in expression of U3. So U3 SNO expression was reduced in human articular chondrocytes exposed to OA synovial fluid, but we saw the opposite effect when we treated with normal synovial fluid. So we also treated the chondrocytes from human articular chondrocytes, normal ones, with some cytokines and looked at U3 promoter activity. And we found that U3 SNO RNA expression is indeed influenced by catabolic cytokines. So the graph on the left, you can see treatment with IL-1 caused a reduction in U3 promoter activity. Treatment with TNF reduced promoter activity. And therefore, U3 SNO RNA promoter transcriptional activity is reduced in normal chondrocytes exposed to 2 catabolic cytokines, IL-1 and TNF alpha. So overall, this shows that reduced U3 levels in cartilage and chondrocytes are a function of osteoarthritis. So the next step in the plan for the U3 was to determine what happens when you overexpress or knock down U3. So this first slide is showing interference of U3 snow RNAs in chondrocytes. So these are normal human articular chondrocytes following knockdown. And to knock down snow RNAs, we use these things called antisense oligonucleotides. So on the left, the top left, panel A, you can see that with knockdown of U3 that we used, we had about a 25 to 50% reduction in snow RNA expression. So in the chondrocytes, we then also measured some other important genes, and we showed that when you knock down U3, you get a reduction in collagen 2. Remember, this is a really important protein in articular cartilage. We got a reduction in SOX9, the major transcription factor of cartilage. We got a reduction in rRNA2, which is a hypertrophic transcription factor, a reduction in MMP13, and a reduction in IL-6. So U3 ASO overexpression in chondrocytes changed the chondrocyte phenotype. So then we did the opposite experiment. So we used a U3 mini gene for this experiment. So this here on the left-hand side is a northern blot of our mini gene, and this is showing you how much increase in expression of U3 you got. So we call this an ectopic increase because it's not a massive increase, but it's actually similar in scale to how much knockdown we had. So we had about 25 to 50% knockdown. Here we're getting about 50% increase in U3 using this mini gene system. So this in 4 donors, individual donors, we've shown that the mini gene increases U3 expression, and then we again measured some of the important genes. So we saw an increase in collagen 2, a decrease in SOX9, an increase in rRNA2, no specific change in MMP13, but really variable. If we look at the individual donors, that's the problem working with primary cells, and a reduction in IL-6. So ectopically induced alterations in the expression of U3 snow RNA in chondrocytes changes the chondrocyte's phenotype. So then we wanted to have a look at ribosomal RNA expression itself. So if you remember back a few slides, I showed that U3 is important in the processing of the precursor 47S rRNA into 18, 5.8 and 28S. So we thought, let's measure the ribosomal RNAs. And we did this in chondrocytes that we'd already treated with OA synovial fluid. And this is because U3 snow RNA is a rate limiting step in the generation of mature ribosomal RNAs. So what did we find? So the first graph here is just looking at human articular chondrocytes and the expression of ribosomal RNA in those chondrocytes. So these are just non-OA and OA human articular chondrocytes. And you can see that in OA, we get a reduction in 18S, a reduction in 5.8S, no change in 28S in OA. And when we treat normal human articular chondrocytes with OA and non-OA synovial fluid, we get changes in expression of those ribosomal RNAs. Well, 18S and 5.8S, but not 28S. So going sort of one by one, if we add OA synovial fluid and compare it to our control, which is sodium chloride in this case, we see a reduction in 18S and a reduction in 5.8S. So in OA, we get a reduction in U3 and we get a concomitant reduction in 18S and 5.8S. When we treat normal chondrocytes with OA synovial fluid, we get a reduction in 18S and 5.8S. And then when we treated with non-OA, so healthy synovial fluid, we actually got an increase in these 2 ribosomal RNAs. So this graph shows exposure of human articular chondrocytes to OA synovial fluid reduces 18S and 5.8S, and HACs exposed to non-OA synovial fluid increase the expression of those 2 ribosomal RNAs. So ribosomal RNA expression is influenced by altered U3. So then we looked at these ribosomal RNAs when we overexpressed and knocked down U3 itself. So in these graphs, again, normal human articular chondrocytes, individual donors shown, when we knocked down U3, we got a reduction in 18S, a reduction in 5.8S, and actually here we had a reduction in 28S as well. When we overexpressed U3, we got an increase in 18S, an increase in 5.8S, but this was not significant, and an increase in 28S. So ribosomal RNA levels in chondrocytes are susceptible to OA conditions, and now that they are dependent on U3 snRNA expression. So then we thought we'd have a look at protein translational capacity. And the way that we do this is something called a puromycin assay. So we found that U3 regulates the activity of chondrocyte protein translation using this method. So since ribosomal RNAs are catalytically active subunit of the translating ribosome, we investigated the consequences for overall protein translation using this method. So the first graph here is showing non-OA versus OA chondrocytes. And you can see that in OA, you get a reduction in translational capacity of the OA chondrocytes. So next, we knocked down U3 and measured translational capacity. And when we reduce U3, not unsurprisingly, we get a reduced translational capacity following U3 knockdown. And if we increase U3, we get an increase in translational capacity. Now, they're not massive increases or decreases in translational capacity, but if you remember, the amount that we're increasing and decreasing U3 isn't that much compared to some experiments where you're overexpressing certain genes. So U3 snRNA is able to change the chondrocyte's translational capacity, and that is important. Why is it important? Because as I said before, you have this imbalance between making and breaking proteins. So it seems there's some things going wrong with the ribosome which affect in translational capacity and may have a major role in development of osteoarthritis. So U3 regulates the activity of the chondrocyte protein translational apparatus. And we wanted to look into this a bit more. So what we did now was we did some proteomics experiments. So my PhD was using mass spec proteomics. So we applied some of these techniques following knockdown of U3. So this experiment was done in 2017 with the help of Ibeck, who was a PhD student in my group, and Phil, who was my technician at the time. And what we did, we took SW1353 cells, and using ASA, we knocked down U3. And then we looked at the proteome and how the proteome was changing. And we used label-free quantification to do this. We then input those genes that, those proteins that were changing into ingenuity pathway analysis, and then produced some results. And this diagram shows the major findings of that experiment within the proteome. So the changes in those proteins, so red is an increase in protein expression following knockdown. We got a change in expression of proteins that affected expression of messenger RNA, expression of RNA, transcription of RNA, and these proteins themselves have effects on initiation of translation of mRNA, translation of proteins, synthesis of proteins, and metabolism of proteins. So the IPA network showed these changes at the protein level when we knocked down U3 in SW1353 cells. And this confirms a global impact on protein translation by affecting these proteins, which are important in protein translation, by reduction in U3. So we also looked at the secretome in these SW cells. And when you look at the secretome, you get a less complex proteome. So in a secretome of an experiment, you're likely to see things like MMPs, which are a low abundance, which will be masked by proteins which are in high abundance within the cell. So this secretome showed that U3 may affect the inflammatory response in osteoarthritis. And we found, so these proteins were, some of the proteins were changing, and these proteins in particular here are involved in chronic inflammatory disorders. So U3 dependent alterations in the levels of secreted proteins involved in balancing inflammatory pathways may also have a role in OA. And then finally, we looked at whether U3 expression is increased when we use an anabolic molecule. In this case, we use BMP7. So can U3 SNO-RNA and its subsequent ribosomal RNA targets be induced in chondrocytes by the anabolic action of BMP7? So we did this in cell line SW1353, and we also did it in human articular chondrocytes. So again, we used a U3 SNO-RNA promoter reporter in SW cells and in the hack. We tried two different concentrations. So in the SW cells, we tried 10 and 30 nanomolar, and compared to control, we showed an increase in U3 SNO-RNA when we add BMP7 to the human articular chondrocytes. And in human articular, in SW cells, and also when we repeated the experiment in human articular chondrocytes by adding BMP7, you get increase in U3 SNO-RNA promoter expression. We also looked at the ribosomal RNAs. So on the addition of BMP7, causing an increase in U3, we got an increase in 18S, 5.8S, but not 28S. And in human articular chondrocytes, again, add BMP7, we get an increase in U3, an increase in 18S, 5.8S, and no change in 28S. And then finally, we looked at protein translational capacity. So by adding BMP7 to SW cells, we showed an increase in protein translational capacity. So BMP7 induces U3 gene expression and increases the expression of ribosomal RNAs. So BMP7 mediated induction of U3 and rRNA increases the chondrocyte's translational capacity. So yeah, a potential way that we can alter this process. So in this part of our talk, I've shown that U3 SNO-RNA, which is important in ribosomal RNA processing, affects the ribosome, affects translational capacity, potentially preferential translation, and changes the cell phenotype. So this study is the first to show the involvement of a SNO-RNA in articular chondrocyte biology. OA-related impaired expression of U3 has important consequences for articular chondrocyte protein translation, rendering U3 a potential therapeutic target in OA. And U3 expression can be induced by the growth factor BMP7, again providing a potential way of targeting U3 in OA chondrocytes. So the next thing I'm going to talk about is the effect of ribosomal RNA post-translational modifications. So in this scheme here, we're looking at canonical SNO-RNAs that affect PTMs on rRNA, and these have effects on preferential translation, translational fidelity, and translational capacity, and ultimately, therefore, on the cell phenotype. So in the second array that we did, we used 30 samples. So the first one we did 6 and 6, and that was young versus old OA. Sorry? Oh, I think somebody's not muted. And in this second study, we used 30 samples, and these samples were from young normal, old protected, and old non-protected. So OA and not OA, or damaged cartilage and non-damaged cartilage in an OA joint. But just to give you a little bit of a background of ribosomal RNA PTMs, SNO-RNAs that come in two classes, the CD boxes and the hacker boxes, and they cause two different types of modifications. So SNORDs cause methylations, and hackers cause pseudo-uridinations. And the CD boxes, there's 137 of these SNO-RNAs, and the hackers, there's 21 SNO-RNAs within the orphan SNO-RNA category. So what are orphan SNO-RNAs? These are SNO-RNAs that are classified in these two classes, but do not cause ribosomal RNA modifications. We don't know what a lot of these SNO-RNAs do. So within our RNA PTMs, these are some maps here, which were actually made by NASA of ribosomal RNAs. And each of the nucleotides that has a little color, you can't really see this very well, is a modification on that ribosomal RNA. Those modifications are important in the 2D, 3D structure of the ribosomal RNA and how they work. And the snRNAs act with proteins to cause these modifications. So the CD box, or the SNORDs, act with these set of proteins on the left, including fibrillarin, which is important for later, and the ones on the right, the hackers, act with these proteins, and discarin. Again, you'll see the importance of that later. And as I said before, most snRNAs, so the snRNAs I'm talking about now, are expressed within the introns of protein coding genes. So a little bit more detail. So snRNAs are regulatory non-coding RNAs. I've got here 50 to 150, but there's debate on that. Some people class them up to 300 nucleotides. They're important in the maturation and site-specific PTMs of ribosomal RNAs. That's their canonical role, their classic role. They fine-tune the ribosome to accommodate change in requirement for protein production, and that's important in development and obviously in disease. As I said previously, most of them are intronic, but U3, as you've seen, has got its own promoter for independent transcription. And some orphan snRNAs have different functions. So these are not your classical PTM producing snRNAs. And some examples would be SNORD116, which is snRNA that we're studying as well. I'm not going to talk about 116. But some of these canonical snRNAs also have non-canonical roles. So some work we've been doing in our group is within these three snRNAs, 32A, 33, and 35A. And I had a master's student, SEMA master's student last year, Zoe, who worked on these, and these are also classed as oxidative stress RNAs, and they're involved in oxidative stress. And as I said before, some such as U3 and RMRP, the snRNA important in cartilage hair hyperplasia, are implicated in pre-ribosomal RNA processing. So why are these canonical snRNAs important? So some of you might have seen this slide before. I love this slide. So this shows that if you remove a single snRNA in zebrafish, and all that single snRNA does is a single modification on one nucleotide at one place on a ribosomal RNA, it causes lethality and morphological defects in zebrafish. So this study, interestingly, looked at SNORD26, they called that U6, SNORD78, and SNORD44. And I'm not going to talk about SNORD26, but that's also one of the snRNAs that we've done quite a lot of work in. So these PTMs, how do the snRNAs work? Well, the snRNA is like the guide sequence for those proteins to cause the modifications on the ribosomal RNA at a specific place. So you can see we've done quite a lot of sequencing. So these are some data sets which are relevant for chondrogenesis, cartilage aging, and OA chondrocytes that we've done since we started this work. And the one I'm going to talk about, which I briefly mentioned earlier, was this microarray of young, healthy, old OA unprotected, and old OA protected cartilage from knee joints. So what we mean by unprotected and protected is within the knee joint, you have areas that are more susceptible to OA. So when we get the knees, in some cases, you have one of the condyles or one area that is very lightly affected by OA, and the other half of the joint, which is severely affected. In some joints, you get all of it is affected, but unprotected is basically normal-ish, OA cartilage within an OA joint, and protected is the normal-ish looking, but it's not normal cartilage in an OA joint. And this is what we did a microarray on. And we found a catalogue of snRNAs that differentially expressed between the three different groups. And from this PCA plot, you can see that the young samples group together nicely, but the unprotected, so the OA samples, clustered in 3 clusters, U2, U3, and U1, and the protected themselves clustered into 2 clusters, P1 and P2. But interesting, when we went and looked, so I didn't say these are all males, so there's no females in this. When we went and looked at the ages, there was a 10-year age gap between P2 and P1. So these are some of the changes that we validated in an independent cohort of samples, and one of them that I'm going to talk about now is SNORD96A. So SNORD96A was reduced in osteoarthritis, and that SNO-RNA was the only one that we found that targets a change in 5.8S rRNA, and it was at this G75 site. So in the array, we found a catalogue of differentially expressed SNO-RNAs. And so what I did then was I went and looked where in the ribosomal RNA these modifications were. And interestingly, it revealed that most of the modifications, so most of the differentially expressed SNO-RNAs were targeting nucleotides in these really important areas of the ribosomal RNA. So for example, in 18S, they were mostly affecting the decoding center. In 28S, the peptidyl transferase center. But within 5.8S, there was only one ribosomal RNA that was differentially expressed in OA, and that was 96A, affecting 5.8S. And this causes a methylation at this site here. So we went and knocked down 96A in human articular chondrocytes, healthy, and looked at what was the changes in ribosomal RNA, chondrogenic, hypertrophic, and OA-related genes. So when we knocked down SNORD96, as you can see here, these seem to have moved. We got an increase in 5.8, 18S, and 28S. We got changes in some of the hypertrophic markers. We got an increase in collagen and SOX9, and we got changes in inflammatory markers and in proteolytic gene expression. We also looked at the protein level. So for ALP, we looked at ALP activity, and we showed that in knockdown of D96, we got an increase in activity. So we have an increase in gene expression, increase in activity. And for COX2, so you can see COX2, you get a massive increase in COX2 when you knock down D96A. And we saw an increase in COX2 expression, which was functionally accompanied by an increase in prostaglandin E2 in the supernatants of those knockdown samples. And then we did the opposite, we did overexpression. And we found that overexpression altered the chondrogenic, hypertrophic, and OA-related genes. So we did it at 2 different time points, but we're just showing you 48 hours here. So altered expression of D96 in primary human articular chondrocytes can profoundly change the chondrocyte's phenotype and ribosomal RNA expression. So I've just shown you our work on 96A, and those figures are part of a paper that I've just submitted to scientific reports. But we've also been looking at these other SNO-RNAs here, which this is another canonical, these are canonical and about orphan activities. So I've shown that SNO-RNAs affect ribosomal RNA, which cause the post-translational modification of ribosomal RNA can affect protein translation and the phenotype. So the other way of looking at it is actually look at these ribosomal RNA protein translational modifications to confirm it. So if we see a change in D96A, is it actually causing a change in the modification on 5.8S? And you can do that by designing special primers to do it. But what we've done now is we've done a sequencing experiment, and this data is being analysed. I'm just going to show you some of this hot off the press stuff. So just go back a little bit to give you some background. SNO-RNAs don't work on their own, they work with proteins. And the important proteins for methylation for the SNORDs is fibrillarin, and one of the other important proteins for pseudouridinylation is discarin. And when we looked at fibrillarin and discarin expression in human articular chondrocytes treated with OA synovial fluid, we found that OA synovial fluid increased expression, gene expression of fibrillarin. Remember, this causes methylation, but a decrease in expression of pseudouridinylation, which acts with the hacker SNO-RNAs to cause pseudouridinylation. So we have done 2 types of sequencing now to try and map these modifications. And the 2 types of sequencing are ribomethyl-seq and pseudouridinylation-seq. And this is in conjunction with Tim Welting's group and a group in Nancy. And so the ribomethyl-seq, we found 109 sites in total identified in our model. So these are young donors treated with OA synovial fluid. 5 of those are differentially expressed. And in pseudouridinylation-seq, 104 sites and 7 differentially expressed. So we've got a PhD student who's now looking at our data sets and seeing what SNO RNAs change, and then looking at this model to see, do they map to the actual modification changes? So in this section, I showed that ribosomal RNA PTMs can affect translation, fidelity, and capacity. But what we haven't talked about are these 2 other areas, and these are areas that my collaborator Tim Welting is doing work in. So I'd like to thank the funders. So I'm funded by the Wellcome Trust at the moment. SEMA, who've supported me throughout. They gave me funding for my postdoc bench fees when I was doing my postdoc. The funders from Tim Welting, our group here, George for his help with the mice, a group from Oxford from the Kennedy who've helped me with to learn the DMM model. And these are Tim Welting's group in Maastricht. This is an old picture from one of our lab walks. And this is Tim, and I'm happy to answer any questions. So one question that I have been asked a few times is, how do I get my step count up so high? So this was last night. I live on this mountain here, and this is me and my son and 2 of our dogs, and I do a lot of running. So I have to admit, a lot of those steps are from running. And the other thing is running around after these guys. So we've got 4 orphan lambs, which we're feeding. These are in our front garden. So yeah, so that's how I'm getting my step count up.
Very good. What's that doing to your OA?
My OA? Yeah. I haven't got OA in my knees. That's is what I would worry about. I've got OA, I think, in one of my toes, because I do fell running, and running downhill is not good because you're bending your toes like this. But my knees are fine.
Good, good. So there are a lot of questions, or a number of questions, but there's several of them from the same group. So I'm actually going to ask the people to ask their questions. So George, do you want to start by asking some of your questions to Mandy?
I have forgotten what I have asked.
Well, you can look in the chat.
Okay. So the main thing is for me is 2 things. One is how if you have a SOX9 and RUNX2 modulating U3 promoter, as you showed, these have opposing effect on chondrocytes. How do you understand their function if they are opposite effect?
Yeah, so we've talked through this quite a lot, and we can't come up with a reason, because you can see not just lows, but also some of the genes are doing opposite things, not doing opposite things when you knock down and overexpress. So there seems to be some sort of partition of the effects or dysregulation. It's not black and white. If it was black and white, it would be nice. So if you overexpress, it increases something. If you knock down, it decreases something. But that's not always the case. And we're working primarily in human articular chondrocytes, and they themselves are difficult because obviously each donor is different. So sorry, George, I can't answer that question really.
Okay. My other questions was relating to, it's just understanding how you do this knockdown in D96. How do you design the things to knock it down, and what percentage of knockdown do you get to get these results?
So we use Tim's big brain. So he designs them, and we get Urogentech to make them, and they are DNA, RNA, DNA molecules, antisense oligonucleotides. So they're based on an original paper from some yeast people, and that's how he designs them. And then we get Urogentech to make them, and then we use them at different concentrations and find them best concentration. But we don't get a massive knockdown. We think if we got a massive knockdown, we probably would kill the cells. We haven't really looked at that. But that's how we do knockdown, the overexpression is a bit different, because depending on the type of snRNA you're looking at, U3 has U3 is a bit different because it's got its own promoter, but D96A, which is in an intron of a protein coding gene, we have to design those differently. So we design those so that they are expressed within a protein coding gene that is ubiquitous, and we use beta globin. And this is from a study by a group, I think in Germany, called KISS, who did the original work, and they gave us the reagents that we needed, and that's how we did the overexpression of 96. So it's not as easy as microRNAs to work with. You can't get off the shelf stuff. You have to design your own stuff the whole time.
Okay.
Mandy, can I come back to your first question about answer to George? Because at the very beginning of your talk, you said that there were 7 different chondrocytes.
Oh, so yes, so from that's a single cell sequencing study where they did single cell sequencing and found 7 subsets. So when but
you're not using different subsets. You're just
we're just using the whole lot. So I also said the chondrocytes change within the different layers, but we're looking at all chondrocytes. Chondrocytes almost like a soup. So we take the cartilage and we take all the chondrocytes out and we look at them. Now that paper. There's I think the first chondrocyte sequencing paper is really interesting, but in order to do that, we would need to find markers for those specific subsets of chondrocytes, and then maybe certain chondrocytes are doing one thing and other chondrocytes are doing another. Yeah. And so I guess going back to George's question, that might be what's happening. So the different populations are reacting differently.
Yes, but that specifically re-asked the question about the role of U3 in OA. Yeah. Because if it is more rank X2 that is driving hypertrophy, then I can understand. But if SOX9 is doing it, it means it is pro-repair.
Yeah. Well, okay, so I think, George, if you can think of any other experiments that we could use to maybe decipher that a bit, that would be good.
So one question relating to this is, and this is my final question, is relating to the, do you see any impediment to using BMP7 as a way to cure OA?
So the BMP7 work is primarily driven by Tim, because he's got some peptides, BMP7 peptides that are in, he's got a big grant, translational grant, to look at using BMP7 peptides, specific peptides, to treat OA. So that work is driven by him. I don't really know much about BMP7, I have to say. Okay. But that's why we also looked at BMP7 in those experiments.
So I think Blondine's got a question about BMP7 too. Blondine, do you want to take on next?
Yes, I can. I just have a couple of questions. Great to see the whole work in one big presentation, Mandy. Regarding the BMP7, so BMP7 actually acts through the SMAD158 pathway, which is actually in chondrocytes, it induces hypertrophy, as well as MMP13 and that lot. So I was just wondering whether that could explain some of the link as well, that is directly linked BMP7 activating your U3 expression, and whether it was through the SMAD158 pathway.
So we haven't looked if it's through the SMAD158 pathway, no. And I couldn't tell you, to be honest. I don't know. Tim might be doing that work because he has got a PhD student and a postdoc on his BMP7 project.
The other thing about BMP7, but again, I'm not an expert either, but I think it's used in cartilage repair at the same time. So it's quite an interesting one to look at for sure in chondrocytes.
Yeah.
And the other question was actually relating to the very first part of your talk. So you started by saying that you had a change in the expression of your U3 in the weight-bearing areas, but not the non-weight-bearing areas.
Yeah, in that DMM model that we looked at, then yes.
So that suggests to me there's a mechanical influence as well, mechanical responses.
Yeah.
And at the same time, you also have an effect on your synovial fluid, which is full of growth factors and cytokines, which are also responsive to mechanical loading.
Yeah.
And whether you knew how these could link basically together to get those responses on your U3.
Not at the moment, but we have got we are looking at what is well, we're looking at the OA synovial fluid and the normal synovial fluid. So Tim's got a PhD student who's literally looking at that. Why is it? So he started doing these, I'm sure other people have done experiments, rather than using L1, treat with OA synovial fluid. What is it in the synovial fluid that's causing these changes? So he's got someone working on that, doing all sorts of different studies. Regarding weight bearing and non-weight bearing, the next stage, I guess, would be maybe to look at using your model, maybe looking at using treatment, U3 mimics, and seeing what happens. I don't know. I need to get more money for the follow-on, really.
Yeah, I was coming to that obviously because of my loading model.
Yeah.
Thanks.
Yeah.
Okay. Vanya, did you have some questions still as well? You've got BMP7 as well.
Thanks, Mandy. Just a quick couple of questions. Noticed that your U3 was particularly changed by BMP7 at mRNA level, not so much at the promoter level. Wondering about the stability of U3 RNA and what regulates no RNA stability, whether that would be an interesting thing.
So yes, that would be quite interesting. In fact, RNA, yeah, I don't know. I could do an experiment looking at the one would be the easy thing would be looking at the half-life. So I've done quite a lot of half-life experiments. So Simon, I don't know if Simon's there or not. So we could do it within that context, yes. Okay.
And is there known much about the mechanisms of stability of SNO-RNAs?
No.
No, I guess that would be quite.
So most of the SNO-RNA work is all done in yeast, and they're interested in just the basics of what they do.
Yeah.
And most of it in yeast, a small bit in E. coli, and the only people that do stuff on SNO-RNAs, like functionally in diseases, is some of the cancer people.
Yeah. A question I had about U3 as well, I'm not sure whether I got this correctly, is when you knock it down or overexpress it, it seems to have the same effect on SOX9.
Yeah.
So just made me think about the clock there, because obviously,
okay.
Just because sometimes when you knock things down or overexpress them, and when they seem to have the same effect, that might be a time of day balance important there. I don't know whether you've ever looked at this one in your time course from the human chondrocytes.
No, that is very interesting actually that you say that. That gives me, we have got some samples we want to look at clock. We've done a clock experiment with some people in with Ian Clark's group. So yeah, that would be quite interesting.
Yeah. And my final question is, did I get this right that the D96A actually goes down in OA?
Yeah.
So, and when you knock it down, all these good ribosomal RNAs were going up.
Yeah, have a look.
So is that, so the question is that a possible compensatory mechanism in OA in order to improve the situation?
Yeah, I don't know. That's a good point. And again, that's a very little bit of work that we've done. And there's so much more that we need to do to find out what's going on.
Okay, thank you. That's great.
I think you just planned your next 10 years of work here.
Well, it's just ridiculous. When you saw those data sets that we've got, which are a lot of data sets, when you do sequencing, you get so much data, and that's just, I'm just looking at a small number of SNO-RNAs within those data sets.
So one more small question from Kazoo, and then I think we'll let you go because you had enough time. Kazoo?
Hi, Mandy, great talk, thank you. Well, this is a very practical question, but people often use ribosomal DNAs you analyzed to usually normalize expression data. Well, so which DNA or genes are you using to normalize those expression data?
We use cyclophilin D generally, but with every sequencing experiment that we do, and if we're going to validate, we look at a set of genes that aren't changing in your sequencing experiment, and then we will check at least 4 or 5 of those and then choose the most stable.
Okay, great.
So I would say never use like 18S for normalization, full stop.
Okay, good to know.
Because we know, the thing is, I've just shown you U3 changes in cartilage, but we know U3 changes in muscle, in aging, and we know U3 changes in tendon, in disease. So they're the other things that I've looked at. And it's really important. So I wouldn't be using any rRNAs for ribosomal proteins, yes, but not 18S or 28S that a lot of people use.
Great,
Well,
thank you.
Thank you very much, Mandy. It's really, really good to see it all together. And I think it's generated a lot of discussions, and I think there are some opportunities.
More questions, though. More questions than answers.
Well, that's okay. That's what it's all about, and what to do next. So that's really good. Thanks, everybody. And again, next week we'll be here, same time, same place. Look forward to seeing you all then.
Thank you.
Thank you.